Our next speaker is Professor Ashish Pathak, Principal Scientist in Chemistry, Drug Discovery Division, Southern Research, Birmingham, Alabama, USA. I am happy to share that Professor Ashish Pathak is alumnus of this department. He has received his doctorate in chemistry from University of Lucknow in 1993. His main research interests are medicinal chemistry, approaches in the area of anti-infective and cancer, carbohydrates, and saponins as immune stimulants, and vaccine adjuvants. Professor Pathak is also an adjunct professor in the Department of Chemistry at the University of Alabama at Birmingham, UAB, USA, and is member of Center for Neuromolecular Research. This is a very brief introduction. I can talk about him much more since I worked with him one time. So could I request Professor Ashish Pathak to please give your talk? Thank you.
Hello, everybody. Good morning. Can you hear me?
Yes,
clear?
yes, you are very loud and clear.
Thank you very much, Professor Khare. We had a good time here in Birmingham. So today I'm going to talk about the work we do here at Southern Research in collaboration with several universities developing antiviral drugs at this moment. So my title of the talk is Antiviral Medicinal Chemistry from Hit to Lead. We work very closely with the UAB School of Medicine. So in this first slide, I'm going to talk about, so this is the Center of Excellence for Translational Research. It's funded by NIAID, NIH. The main is Antiviral Drug Discovery Development Center. That's what is called 83C. And the main PI is from University of Alabama at Birmingham. Then there are 4 projects in this whole center. One belongs to flavivirus, and the PI is from Oregon Health Science University. We work on antivirals for dengue, West Nile, and Zika. The second project is coronavirus. We work on SARS and MERS, and the PI is from Vanderbilt University. Alpha virus, we work on Chikungunya and VIV. The PI is from Oregon Health Science University, Oregon. And influenza, we work on 3 different strains of influenza, H1N1, H3N2, H5N1. And the PIs are from UAB and SR. I think it's going too fast. Okay, so looking at what my role is, so I am the co-director of medicinal chemistry. High throughput screening assay core is at Southern Research too. And then we work very closely with Emory Institute of Drug Development at Atlanta. They have also the medicinal chemistry and lead development core. Our industry partner is Gilead. So first I'm going to talk about what are our contributions to SARS project, coronavirus project, and especially SARS-CoV with this epidemic. So we looked at different compounds 4 or 5 years ago for SARS-1 epidemic that happened in 2004. So we were looking at the RNA-dependent RNA polymerase inhibitors for coronavirus. So now everybody knows about this compound, known remdesivir. The synthesis was done by Gilead, and this compound came to the center when they were working on Ebola at USAMRIID. So we took this compound for coronavirus, and that's the coronavirus 1, basically. That's what I can call that, SARS coronavirus. And then we tried to develop this compound for coronavirus infections. So this compound was active against SARS-CoV. It was active against MERS-CoV. MERS-CoV comes from camel and is more prevalent in the Saudi Arabia area. And this compound is basically a proteid, and this doesn't have a natural nucleobase in there. This is a proteid because this is a monophosphate form. So when this compound goes into the body, it converts into monophosphate by removing this protecting group over here, which is the rate limiting step. So if you take a pure nucleoside, that's the rate limiting step to make the monophosphate. So we give monophosphate in there, and then it converts into diphosphate and triphosphate. So it converts into monophosphate by a reaction by esterases, and then monophosphate then converts into triphosphate, which is the active form of compound. How this one works? This one works especially from the evading the proofreading of the viral ribonuclease, causing a decrease in the viral RNA production. And this is a direct antiviral agent that works as a delayed chain terminator. So this was basically developed for MERS. Right now, the clinical trial is going on in Saudi Arabia on this compound. But when the SARS-CoV came in, this compound was taken for SARS-CoV and developed by our collaborators at Vanderbilt University. And this is an approved drug right now in the United States. The second molecule I'm going to talk about again is RdRp inhibitor. And this is a very recent compound. This compound is known as EIDD 2801. This is nothing but a prodrug of cytidine. And this compound is also active against SARS coronavirus, MERS coronavirus, and this has been tested in SARS-CoV-2. Recently, clinical trials have started in UK on this compound, and this compound came from Emory Institute of Drug Discovery into our center. So as I said, this is a 5' ester product of N-hydroxycytidine. So if you look over here, instead of an NH2, it has an NHOH. And it converts to N-hydroxycytidine, which goes through the monophosphate, which is a rate limiting step. Again, this ester bond is going to be clipped off, and then it forms a monophosphate and then makes a triphosphate. This exerts its antiviral action through the introduction of copying error during the viral RNA replication. And again, this is a direct antiviral agent that works as a chain terminator, and we have high hopes that this compound will be successful in UK trials and can be approved for SARS-CoV disease. So now I'm going to switch a little bit gears. There are so many other efforts are going on. We have some small molecules in SARS-CoV area, which I cannot show here. We have already performed several high throughput screening. We have a lot of compounds that we are working right now. We also screen the FDA library. So I'm going to talk about how we work on these antiviral agents and what is the role of this core, the medicinal chemistry core, and what exactly we do. So these are the specific aims for us, basically. So we identify screening hits from any HTS screen where we screen hundreds, thousands of compounds. We also looked into the pre-existing hits like MLPCN screen by NIAID. We also looked into our own screening programs. We optimize chemical hits for antiviral activity through medicinal chemistry. So basically, we optimize the pharmacokinetic properties warranted for animal testing as a proof of concept first. And we also provide integrated informatics support that includes compound tracking, data analysis, data storage, backup, and retrieval. So it's a very big program, and there is a lot of people are involved in this. So this slide basically describes here what is the hit to lead chemistry. So what is the paradigm of hit to lead chemistry? This is adopted from this publication from BMCL. So if you look at it, there are different ways from where you can get some molecules that you can optimize for activity as well as drug-like properties. So main area is the high throughput screening. This can be a biochemical or a cellular screen. We also look into in silico screening, which is also called as virtual screening, where there are the models available for proteins, and a lot of commercial libraries get screened, and based upon their interaction with the proteins, the compounds are selected based upon their energy in kilocalories. There are some focus screens, like there are some antiviral compounds are available through commercial sources. So we have sets, like we have kinase set, antiviral set, antibacterial set. So we use those specifically for a specific project. Then there is a phenotypic screen, which is cellular. And then there can be a fragment-based screen where we identify compounds in their molecular weight range of 100 to 120, and then we grow the molecule based upon the structure. So from there, we get the apparent hit. Then we remove it through the Payne's filtration, and the hit quantification is done on this. That gives us the true hits And then we start the hit to lead chemistry to get the quality leads, and then we optimize those compounds for quality candidates. So what are the libraries at Southern Research? So we have about 1 million small molecules plated in 384 well format, and they have been assembled from different commercial libraries named like Cambridge, Enamine, Tripos, Life Chemicals, etc. We have a very specific kinase library that has about 26,000 compounds. Other than that, we have approved drugs and biological active libraries. We have about FDA library. We have some unapproved drugs which are in clinical trials right now. And then we recently acquired a reframe library from Script, which is about 13,000 compounds through Gate Foundation, which we have screened for SARS-CoV, and we have very good data from there. And we are moving forward with a lot of compounds. And then we have small fragment libraries of 20,000 compounds. In addition to all this, SR has a big chemistry division, and we produce a lot of compounds every year, and we assemble that into a library also. And we have more than 40,000 compounds, which were synthesized in-house. So what is the general strategy for lead identification? So we start doing an HTS screen. It can be, as I said, it can be a target-based screen or just a cellular assay. We determine EC50, that the inhibition of the compound, effective concentration at 50%. And then we also do CC50, which is the cytotoxicity in the primary screening cell lines. After that, we do a clustering analysis. So basically, we try to see how many compounds of the same scaffolds were active. That gives us the confidence in that particular scaffold to move forward. And then also we do a PAINS filtration. PAIN is nothing but a pan-assay interference compound. So there are about 400 structural classes that are not drug-like. They have some issues, either a toxicity or some binding. So we try to remove those very early before going to the reconfirmation hit. Then we do the primary assay, or we select a SAR assay, which is a structure-activity relationship assay. And as I said, we do a cytotoxicity in the primary cell line. From there, we select hits. So our cutoff value, for example, for this program is less than 15 micromolar EC50, greater than 30 micromolar CC50. And in parallel, we also do the viral titer reduction assay. So this is very important, because if the compound is not reducing the virus load, or called as a virus titer, then the compound is going through a different mechanism and it's not very effective. We choose compounds from there, and then we do in vitro ADME properties on it. So basically, we look at the microsomal stability, solubility of the compound, and the log D. That gives us a way of choosing a better compound to start with. And then we do SAR assay, do cytotoxicity in parallel. That gives us the lead compound that goes into in vivo PK in the animal model. And most of the animal models we have here is mouse model. And that gives us a lead compound to move forward. So I'm going to give you a HTS screen. We have performed a lot of HTS screen, but this is one particular screen I would like to share with you. It's the Chikungunya. Everybody knows in India what is Chikungunya virus. And we are working on this for last 4 years, and we have several good compounds which is moving forward in primate models. So we started with 197,000 compounds using cell titer glow luminescent cell viability assay. So CPE assay, basically what we look at is we look at the health of the cell, how we can protect the health of the cell which are infected. So it's an indirect way of looking at it. So we are not looking at the virus reduction, we are looking at the health of a cell rescued by these compounds in presence of virus. So for example, from there we got about 2,558 compounds, and those were actually went through clustering analysis and PAINS filtration. We selected about 44 compounds, and 23 we purchased for reconfirmation. So every time we have some hits, we generally go back and buy commercially a fresh powder, look for their integrity, structural integrity, purity, and then reconfirm those compounds, and then send to virus load reduction assay at 10 micromolar. And then we try to determine EC90. So EC90 is basically 1 log reduction of the virus, which is very important. And generally, our cutoff for a compound is greater than 3 logs at 10 micromolar. And from there, we have 5 hits selected for lead generation, and we are working making more compounds. So here in this slide, I'm just going to share with you that this is a reporter virus we created for BSL-2 level. And this here in this first panel over here, you can see that this is the antiviral effect of a compound. And on the right-hand side here, this is showing the toxicity of the compound. So looking at what we try to do in SAR basically. So in SAR, as you can see, we can have a target potency, we can have a receptor drug interaction, modification, modulation in metabolism. We look at the bioavailability of the compound, we do scaffold hopping and so on and so forth. So this basically cartoon shows here that what kind of properties we look when we do a SAR analysis. So SAR structural analysis, first we try to purchase any close analogs from the commercial sources, and if there are none, then we try to synthesize those compounds. All analogs needs to go through efficacy, selectivity, and toxicity analysis. Data used to feedback to generate more analogs. So as we go forward, we make more compounds based upon the previous data. And the promising compounds from there usually go in ADME properties. So where we look at the solubility, aqueous solubility at pH 7.4, we look at the microsomal stability and log D. And then we take the good compounds into in vivo PK, where we try to find the T-half, the half-life of the compound, what is the maximum concentration in vivo, and percentage bioavailability. So these are some of the properties. We look at it very carefully. So molecular weight, polar surface area, we look at the solubility, log D, which is the distribution coefficient. We look at the partition coefficient, log P, which is a calculated value. And we look at the Lipinski rule of 5, where we look at these properties and select compounds from there. This is also very important. We look at the mouse liver microsome, rat liver microsomes, and human liver microsomes for the stability. As you can see, there are drug metabolizing enzymes, which is called CYP iso enzymes, and several of these enzymes we test. And generally, this is very important to move a compound forward into an in vivo model. And we also look at the hepatocyte activity in mouse, rat, and humans. And generally, we look at 120 minutes T-half percentage of the compound remaining, because these are the liver cells, and these are going to change compounds in the liver. So how we increase solubility, for example, so we try to add some polar solubilizing group on the molecules. We try to add some cycloalkyl molecules, insert heterocycles, interrupt the intramolecular hydrogen bonding, insert groups that will allow rings to rotate with the molecule. So we disturb the planarity. That's how the solubility is increased. If you look at the structural alerts, so there are so many data available that these kind of moieties are structural alerts. We try to not use those into our molecular designing, and that's how we increase the metabolism. We try to use some bioisoesters, basically having the same kind of properties, block sites in the metabolism, especially in the aryl rings. And this is one of the software which we use a lot, which is called CSL value. It's from StarDrop, and that actually helps us a lot looking at the liability sites in the molecule where we need to do changes to stabilize the molecule. And this is basically a publication I took out. So we get this kind of a data here that tells, so red is basically is metabolizing very fast. So if you look at this molecule, this methoxy over here is a 51% liability, and this gives us a CSL score. So any CSL score which is less than 7 is a good molecule to move forward. So these kind of analysis we do to check the cytochrome P50. So for every drug discovery program, we have a profile. So this is the lead profile, which is applicable to all the viruses we are working on. So we are looking for a EC50 less than 0.25, EC90 of less than 1.0, cytotoxicity, molecular weight. So we plan these things ahead of our moving forward with the SAR. So basically this cartoon over here describes what are the medicinal chemistry strategies. So basically here you can see the active compounds come into this cycle over here where we synthesize, we test, and then we do the in vitro ADME and try to optimize the compound so that we have a lead compound with these target values. And that goes into a secondary assay calculating the EC90. And if we get a good EC90, we do an in vivo PK where we try to make an oral compound with a T-half greater than 2 hours and percentage F, which is the bioavailability greater than 30. So all these things goes back. So if something happens here in vivo PK, we go back, we try to optimize compound again, and then it comes in the circle. Then compound goes to murine model, and if the compound is not efficacious here, there is some issue, we go back and we try to do changes so that the compound is more stable, for example, or is going. And these are some of the secondary toxicity assays we do. Once the compound is clear from the murine model, then we try to look at the plasma protein binding, hepatocyte stability, CYP, enzymes, and mini-AIMS. HERG is the heart problem, and selectivity profile of the compound, and that goes to a preclinical candidate. So this is the whole chart that we use. So what is the right balance to take a compound forward? So this cartoon here tells that we look at the potency selectivity, we also look at the solubility, we look at the bioavailability, and if the compound has to go into the brain, then the brain penetration, and all these properties we try to balance the optimized candidate to move forward into in vivo efficacy for proof of concept. So I'm going to give you one example over here. So this was one of the hit 33366. This compound had a EC90 of 3.2 micromolar, which is above the range, has a solubility of 5 and a very poor microsomal stability. So we start chemistry on this. The first thing what we did is we changed the O-methoxy and did some alkyl substitutions, aryl substitutions here, and tested those compounds. We also reduced this amide bond to amine substituted. We also changed the size of this ring, as well as we changed both at same time. About 80 compounds were synthesized, giving us this 7-membered compound number 34963. As you can see, we have decreased the EC90 tenfold. So the compound is more active now. The compound is decently stable in microsomes, is 47 and 50. So we took this compound and moved forward. So this was our starting point, and then we started doing changes, and the major changes we are trying to do is increase the solubility and making all these analogs of different substitutions. We came to one compound which is known as 36498. Its EC90 went up, but as you can see, the MLM, HLM has gone up too. So this was some of the SAR properties, and as you can see, this is nothing but a reverse amide of this compound. So basically just reversing an amide can improve the metabolic profile. And from there, we made more compounds, do about 65 analogs we synthesized, and then we came down to this compound 39689, which was our candidate for in vivo efficacy studies. As you can see, the metabolicity is very stable. Solubility was very low, but for proof of concept studies, this compound we were able to formulate to test in vivo. So this is the profile of this compound. As you can see, the EC50, EC90 values are less than 1 micromolar. It has a very nice VTR greater than 3 logs here. It has a good MLM value. It has a decent clearance. HLM is 300. Solubility was a factor which we are working on. And this compound also shows activity against VEEV, which is another alpha virus. It shows activity against West Nile virus, and it also showed activity against dengue. So this is a multi-virus active compound, so our interest is there to move this compound forward. So we formulate this compound in 15% DMA and 85% PEC 400 for pharmacokinetic analysis. We did 4 different routes: IV, PO, IP, and SC. So PO is the oral route, which is our preferred route. But this compound, for proof of concept, we thought that it's good to take this compound IP because that's where we got the maximum concentration. So as you can see, this is about 2.4 micromolar concentration. We got a percentage F of 43. So and T-half is 18.5 hours. So we are taking this compound forward. This data actually shows here because chikungunya at the latest stage, it goes into the brain also. So we would like to see whether our compound is going into brain. So as in this table over here, you can see what is the plasma concentration up to 8 hours. You can see how the brain concentration in 8 hours, and you can see that it's increasing in brain. However, it's decreasing in plasma, which is very natural. And here you can see that the brain to plasma ratio is increasing. So that means this compound is going into brain and accumulating in brain. So basically, this will be an effective compound moving forward. This is the toxicity study we did. We took 20 milligram per kilogram. BID is 2 times a day, IP injection, 5-day dosing, male and female mice, 100 microliter injection volume of the formulation, and we did not see any toxicity. And then we took the tissues from these mice. We took his brain, spleen, kidney, liver, ankles, and quads, and those samples were analyzed for compound. As you can see here, this is the female 20 mg per kg dose. Here's the male 20 mg per kg dose, 40 mg here. As you can see that this compound is available in every organ we took out. So basically this compound is very nice in different tissues. This is the mouse model we are using to evaluate this compound. And as you can see, this is the 10-day model. And here in this graph, you can see that compound is in 3 days, basically the viral reduction, the first storm comes and then it goes down. And then again, the second viral storm comes in 7 days and 8 days. So it increases there. And that's where the cytokine storms come. And that gives the arthritis kind of a symptom to the patient. So we are looking at this compound into this model, and we are making more compounds, better soluble compounds. But this is a proof of concept model which is ongoing. So just to give you an idea, in 5 years, basically we produced about 1,500 compounds synthesized for six different viruses. 25 hits to lead campaign were done, and especially these are the flavivirus, coronavirus, alpha virus, influenza virus. 16 leads were optimized studies and still ongoing. 5 leads were taken into in vivo PK studies, and 4 leads are taken into animal efficacy studies. And we are moving forward with several compounds in primate model. This cartoon here shows that what are the capabilities of Southern Research. Basically, we do lead optimization, proof of concept animal model, and we have all kinds of chemistry integrated with our electronic notebook system. We have computational, we have structural studies. So basically, we have a complete drug discovery platform to carry out from hit to lead. With that, I would like to thank the whole group. So this is the medicinal chemistry group. As you can see, this is a big program, a big effort from a lot of people. We have a structural biology group, in vitro ADME group, computational chemist. We have compound management, and some students are trained also. And this whole work is being funded by NIAID through these 2 grants. Thank you very much. And if you have any questions, feel free to ask.
मेरी साइड पर
सर ये अभी आपको जो इनवाइट करेंगे ना सर, आप ऐसे नहीं होगा, फिर इसके शेयर स्क्रीन पे सर जब आप प्रेजेंटिंग करेंगे।
मेरा का टाइमर लगा देना सामने, टाइमर लगा दे।
Ashish, there is a question by from Tuba Kamar. My question is for Ashish sir, how can we control the rate of mutation of coronavirus?
So rate of the mutation you cannot do, because that's natural process in viruses. Basically, it goes through several different ways of mutations. Sometimes the lethality goes up, sometimes it goes down. So at this moment, what we are seeing is there are about 40 different strains of SARS coronavirus floating around. Some has a higher infectivity as compared to the other.
There are a couple more questions. Let me find out.